So the course that I work on is the Fundamentals of Neuroscience, and you can find a lot of our stuff. We also have like a Facebook page. I have my personal Twitter, but we also have our own Twitter account where you can tweet questions to us. And what we really were aiming for when we created the course was to just get people really excited about neuroscience and to feel like they had some sense of agency. Like, I can be a scientist. I could be a neuroscientist too. I can learn some things and maybe teach some other people things about neuroscience. So that was like our big goal with this course. And we offer a bunch of different types of things on the course, and in the course. So we have kind of like standard lecture videos, although they're much shorter and much more condensed than like watching a professor, at the board and a chalkboard talk for an hour. We took standard lectures and we rewrote them entirely, so we kind of scripted them like a movie to try to make them as condensed as possible. So they're pretty short, but there's a lot of information in them. You can probably attest to this. Some of them you have to watch over and over again because there's a lot there. We also have really cool science documentaries. So my background is also in documentary filmmaking, and so I really tried to make the course as exciting as possible, and that means doing things like shooting a deep brain stimulation surgery and like going into the life of somebody who has Parkinson's and thinking about and asking them questions about what it feels to go through that. We also go into labs of different neuroscientists in and around Cambridge, where we're based. So we recently went into the lab of this really awesome neuroscientist, Kay Tai, who's at MIT, who is working using optogenetic techniques. So I don't know if you guys are familiar with that, but basically you have lasers and you can shine them on neurons and turn them on and off, which is crazy because you can have a live rat running around and within a very short time scale, seconds, you can turn on and off very, very small regions of their brain, which before we could do very roughly using like chemical techniques or like physical techniques, like taking out a piece of brain via surgery, but it was pretty rough, and now we're getting down to like very, very, very high resolution. So we go into her lab and she kind of shows us the whole process of how do you do that from like, unfortunately, here's the mouse and here's its brain, to this is what we're looking at through a confocal microscope, and you can see some of these neurons are stained and the others aren't, and this is what it behaviorally looks like. So we do kind of cool stuff like that so you can see what's actually happening. We also have really pretty animations that we tried to make so that we could get as many people who like have no interest in science at all to check out the course and come in and get excited about it. And we have some virtual labs, which I'll show you, which are basically like some of the lab that we're going to do today. We have a virtual version of it. So if you want to play around and change some of the parameters and physical constraints in a lab prep, we make those. And we're building those up. As of now, we have about 7 or 8 of them. We've put out 2 modules, and so these are really awesome tools for learning some of the more complex biophysics of what's happening in neurons in the brain. Then we also have a DIY lab section where we have videos and lots of educational materials, so if you want to do labs using a spiker box, we have a bunch of those and documentation, and we filmed Greg Gage, who's one of the founders, and he came in. So there's a bunch of material there. And then we also have a forum community, and if you want to find answers to questions, this is a bit of a tricky one for us because there's so many people who take the course, and so at the moment we use Piazza, which is a type of forum which is okay, but not the best, so it can sometimes be hard to like find what you're looking for, but that's what we have. And also a lot of people who are working on the DIY labs on this forum post their problems and their results, and you can kind of engage with them or see what other people have done. We also have exam periods, one of which is actually happening right now. So these open exam periods are for each of the modules in the course, and when you take those exams, if you pass them, you can get a certificate with edX that you passed the course. And we offer those like every 6 months or so. So there's a lot of stuff there, but it's all there for you all the time anyway. And I can show you some of what that looks like. Do any of you guys have any questions about that? No? Okay, so this is our website, and you can see here we have different modules. So this is the first module, and the experiment that we're going to be doing today is related to the electrical properties of the neuron. So in this module, we have 4 lessons which relate to the resting potential in a live neuron, and what are the properties of that membrane, including things like channels, and the ion content of the intracellular and extracellular fluid that give rise to a potential across the membrane, so a difference in voltage. And then we talk about what an action potential is and what action potential propagation is. A nice little virtual lab which shows you what happens when you're injecting current, so this is like a fake action potential, into a cell. And what's happening with your action potential, which is this spike here, which is the potential between the inside and the outside changing from negative 70 millivolts, which is in a mammal, usually the potential of the cell, jumping up, depolarizing, and then dropping back down again. And what's happening with these channels that are letting in different amounts of sodium and potassium. So we have lots of different virtual labs like this, and then you can see actually what happens to, what happens to the action potential when, for example, you eat some bad pufferfish and you block your sodium channels. So if you don't allow any sodium to go into the cell at all, or go out through these active channels, this is what happens to the action potential. There is none, okay, because you can't change the number of positive ions versus negative ions on the inside and the outside of the cell. Like this one, which is a way to explore how the membrane potential changes when you have an axon which is myelinated. So, do any of you guys know what myelin is? Yes?
So between the terminal at the end and the dendrites, there's basically something called a myelin sheet, and that's basically a covering of the Schwann cells that connect that area.
Yeah, and it's basically like a form of insulation.
Yeah.
So they're made out of mostly lipids, fats, and they're totally insulated. Yeah. So it's just a sort of way around that.
Multiple sclerosis is when you have the demyelination of the...
Yeah, so when you have damaged myelin, so if you have a current going down and it's jumping from node to node, and that's what's happening here, you have the membrane potential jumping from node to node. If you have damaged myelin, it basically stops it from jumping because you have this diffusion, so you can see what happens here. You see it just trails off. That's the membrane potential, and it's trailing off, and it basically is not able to jump. It's not able to create enough of a differential by the time it gets to that node to jump to the next node. So these are kind of fun ways of playing around with that. We have a whole bunch of other ones like this, and the nice thing about this is even for, as we were creating this course with a lot of other neuroscientists, they forget a lot of this stuff. You don't necessarily think about this when you're doing high-level experimentation, but it's really important to learn, and these are kind of nice ways of doing it. So this is one of the videos that we had from our most recent module, and so the first module, which I showed you before, which is up here, is the electrical properties of the neuron, and the one that we just finished is called Neurons and Networks, where we talk about how you learn, what potentiation and repression are, and how neurons are modulated when they're speaking to one another, basically, when they're sending signals back and forth. And so for one of them, we made this pretty cool video. That's Dave. That's not the cool one. No, that's cool too, but not as cool as this one. So we worked with an animator from the Royal College of Art in London, and she made this really beautiful animation with us, talking about action potential propagation and the synapse. So, do you guys want to see this? Yeah? Okay. In the last module, we took first steps towards understanding the electrical properties of individual neurons. We learned how electrical forces and diffusion give rise to membrane potentials, and we learned how cells can generate and propagate signals called action potentials, or spikes, along the membrane. Understanding the properties of the neuronal membrane is essential. But understanding just these properties isn't sufficient to give us insight into collective behavior of the billions of connected neurons in our brains. Luckily for us, we can approach neuroscience at many different scales and levels of analysis, and we don't have to confront the full complexity of everything all at once. That's what we'll be exploring throughout the rest of this course as we slowly go from our understanding of single molecules, such as ion channels, to the electrical behavior of neurons, to their collective behavior in small circuits, and finally onto how they become organized in large functional regions of the brain. Let's start simple though. Since we've examined one neuron, a logical next question is how do two neurons connect with one another? We'll first examine some basic cellular anatomy of neurons. So far, we haven't made too much of the fact that the majority of neurons are polarized cells. That is, they have one portion of the cell for receiving inputs and another portion for sending outputs. The parts of the cells that are specialized for receiving inputs from other cells are called dendrites. The word dendrite comes from the Greek word dendron, meaning tree, and as you can see, the dendrites have a branching, tree-like structure. A signal received by a dendrite is passed to the cell body. If there is sufficient depolarization of the cell body membrane to initiate an action potential, then an action potential is sent down the axon. The axon then carries the propagating action potential to another neuron. So what actually happens at the boundary between two neurons, between the axon of one neuron and the dendrite of another? This interface is called a synapse. There are two general types of synapses: electrical synapses and chemical synapses. Electrical synapses are less common in our own nervous systems, but they're simpler to think about, so let's start with them. Electrical synapses are basically pores between two cells that allow ions to pass through. They allow the passage of the electrical signal through to a neighboring cell without much fuss. It's not so different than just combining two cells into one larger cell. There are lots of reasons that nature might need synapses like this from time to time. They're fast, and they allow cells to couple together with a high degree of synchronicity. But most neurons are connected together by a much more complicated structure called a chemical synapse. In a chemical synapse, rather than simply passing along an electrical signal from one cell to another, the action potential travels to the end of the axon and causes a chemical to be released into a very small space between the two neurons called the synaptic cleft. This chemical is taken up by the downstream neuron on the other side of the cleft. This chemical signal can cause the downstream neuron to depolarize its membrane, converting the chemical signal back into an electrical one. Or it can have other effects on the cell. This chemical step is slower than transmission across an electrical synapse, but it opens up an enormously diverse repertoire of different and more complex kinds of signaling. And synaptic function plays a critical role in computations performed by neurons. We'll spend the rest of this unit exploring the inner workings of chemical synapses, on our way to beginning to look at how networks of interconnected neurons give rise to behavior. We'll also look at the role of defective synaptic physiology in neurological and psychiatric disorders, and we'll see how synapses can be targeted by various psychoactive drugs and poisons. Finally, we'll wrap up by looking at how synapses can change over time in response to external stimuli, playing a foundational role in how we learn and remember. So that's like a little... I think what we're going to do now is try out an experiment which actually has to do with the electrical properties of the membrane. So we're mammals. We have mostly these myelinated axons, the guys that we're going to be playing around with, these cockroaches, are not mammals, so they have unmyelinated axons. Their leg looks like this, and it has these little barbs on it, which are basically sensory organs. So when a cockroach is scuttering around on the floor or wherever it is, if it touches things, there's a mechanical reaction that happens actually with these little barbs, and it induces a spike train of action potentials and tells the cockroach that something's touching its legs. So this is a really nice way for us to basically induce an electrical current, which we can measure by sticking in these electrical pins into two parts of the leg. So I'll leave this up here for you guys to look at, and there's also some little cards that you can look at too when you're placing the pins. The cool thing about invertebrates is that their axons are really big because they're not myelinated. So you either have very small myelinated or insulated axons, or you have like really huge ones, and theirs are pretty big, which is why we can use these big pins. In an actual neuroscience lab, when you do electrophysiology, you have, and you're trying to measure or record axons that are myelinated, you use super, super tiny pins, essentially. So this is nice because we don't have to do that, and it's not as expensive, and it's great. And basically what you're going to do is you're going to do this, you're going to turn on this microbox, which is just an amplifier, essentially, and it's turning that electrical signal into a signal that you can hear. And if you have the app on your phone, you can also, are there any splitters by any chance that you have here? Like an audio splitter, you know,
Oh, yeah.
if you do have them, that's fine. If not, you can either listen or see them. So you can listen and hear the crackling, and when you touch these barbs, you can hear the crackling change, and that's lots of different spikes happening. Or you can plug it in and actually see the spikes on your screen. So we'll do that one first, and once everybody's had a chance to kind of try that out, we're going to try the inverse, which is where we put an electrical signal into the leg and see what happens to the leg. Okay.
For 5 minutes, maybe?
Yeah, 5 minutes, and then you can all meet me back here, and we'll do a little wrap up.
Yeah, I did go with methodology here. It's in a lot of units. But use what you want. It's very similar to methodology. You need it.
Variant of light. No, it's so I don't know much about it, but I know that you are here.